Hello everyone, I am Matteo Baldelli and welcome in our video presentation. In this presentation, we will talk about electroosmotic flux in a completely uncharged solid-state nanopore using an atomistic simulation approach. Thanks to advances in technology, nanopores have emerged as a powerful tools for several global challenges such as nanopore sensing, blue energy, water desalination and filtration. However, the technological advancement in nanopore systems are hindered by our lack of knowledge of all the phenomena that occur at the nanoscale. One of the most important challenges is understanding the coupling between electroosmotic and ion fluxes. For this reason, our work is focused on the study of the coupling between electroosmotic and ion fluxes in a completely uncharged and high confinement nanopore system. Our general simulation system setup is composed of an uncharged cylindrical nanopore built using a Lennard-Jones solid, immersed in a 0.6 molar concentration of sodium chloride aqueous solution. The simulations were performed for nanopore radius in range 1–2 nanometers and from different applied voltage, from -0.25 to 0.25 volt. Let's start our result discussion from nanopore with a radius of 2 nanometers. If we look at the fit line of the electroosmotic flux, we can see how the line lies on zero value, and also zero is always inside the error bar. If we compare this with the value of the ionic current ratio, that is the ratio between cationic and anionic current, we can say that the pore is not ion selective, and then electroosmotic flow is zero. A different scenario occurs for smaller pores. Here we have a nanopore with a radius of 1.3 nanometers taken as an example. In this case, the fit line of the electroosmotic fluxes for different voltages suggests to us that there is an electroosmotic flow. In fact, if we compare this with the value of the ionic current ratio, we can see that the cationic selectivity is observed. This result is true for all the nanopores that have a radius smaller than 2 nanometers. In conclusion, high confinement nanopore systems with radius smaller than 2 nanometers show the possibility to obtain ion selectivity and then electroosmotic flux. For larger nanopore radius, the selectivity and the electroosmotic flux are no longer observable. The aim of the future work will be to continue and improve this analysis and study the effect of nanopore wettability on ion selectivity and electroosmotic flow, perform simulation for different pore geometries and different concentrations of the electrolyte solution. Thanks for watching and for your attention.
Hello. Water desalination, DNA sequencing, and blue energy harvesting represent growing industrial demands. These processes rely on the selective and conductive properties of the membranes that are used, which in turn are partly determined by the dehydration barrier arising when water molecules are stripped off or restructured near the ions. These water structures or patterns are routinely reported in molecular simulations, but an analytical description has not yet been attempted. In this presentation, we show that we are able to quantify the hydration patterns using the bulk radial functions. This method has been already used and it stems from the PMF decomposition, and it has been applied to solvation of DNA. Here we extend the existing method by taking into account the presence of a single ion, and we use molecular dynamics simulations to get the reference water density. So what are our results? First, we demonstrate that our theory and the MD simulations agree well. Secondly, we demonstrate that the method is capable of predicting the positions of trapped water molecules. And thirdly, it can capture the intrinsic and extrinsic features of nanopores. Intrinsic features are specific for the pores themselves, such as geometry, size, isomers, number of layers, and their offset. Extrinsic features are controlled from outside, such as strain, bending, and twisting. So, by choosing a nanopore and by controlling by tuning its properties from outside, one could engineer the hydration pattern around an ion. How can it be useful? First, one would be able to provide a fundamental insight into the dielectrics electrostatics and dynamics of ions in nanopores. But primarily, one would be able to engineer the potential of the mean force, and in doing so, to design and optimize controllable nanoionic devices with on-demand selective and conductive properties. This is of enormous importance for water desalination to maximize the water quality and minimize the energy cost, in energy harvesting to maximize the harvesting efficiency and power yield, and DNA sequencing to improve the precision of the nuclear-based detection via ionic currents. Another potential application is external control over the DNA's translocation speed in order to slow it down. So, to summarize, by engineering the hydration patterns, our method can help to design and control the transport properties of artificial nanopores in biophysics, nanotechnology, and industry. And as a final note, I'm currently looking for a postdoctoral position, so if you've got a suggestion, please let me know. Thank you very much for your attention. Bye.
Welcome to my poster presentation introduction. I'm Dario Calvani, PhD candidate at the Leiden University, the Netherlands. Nowadays, we know atomically thin layer materials with a nanometer size pore are promising candidates for ion exchange membranes. In this context, today, I'm going to talk about my computational work within two exciting joint projects at the Leiden University. Actually, I investigated the supramolecular aggregation of polycyclic aromatic hydrocarbons on the water surface at DFT and molecular dynamics level. Experimentally, the synthesis with a narrow pore size distribution of membranes, however, remained challenging. Starting from polycyclic aromatic hydrocarbons called PHs, a two-step approach, the Langmuir-Blodgett membrane formation, followed by thermal annealing, has been used to synthesize molecule-thin carbon membranes with a tight pore size distribution. Everything starting from two pH molecules types. In the red square, the HPA-HBC molecule, or in the yellow square, the HTPY molecules. My computational focus was on the mutual pH's first contact above the water surface. Using DFT and molecular dynamics simulation, I analyzed the orientation of pHs above the water surface, I mean the tilt angle of this molecule. And I found out that this orientation depends on the balance between two intermolecular forces, hydrogen bonds between pHs and water molecules, and pi-pi stacking between pHs molecules. The results from the energy estimation by DFT and from the orientational analysis by MD gave us a main conclusion. Pi-pi stacking and hydrogen bond may be competitive and/or cooperative intermolecular interactions depending on the number and type of molecules in order to form efficient nanometer size pore membranes. If you want to know more about my work, please visit my poster. Thank you for your attention and enjoy the poster presentation.
Hi, my name is Flavio Costa and I am a PhD student in Theoretical and Applied Mechanics at Sapienza University of Rome. Under the supervision of Professor Giacomello, I am studying the ERG-K plus channel gating mechanism. The human ERG channel is a voltage-gated potassium channel expressed prevalently by the Hurst cells. It plays a key role on the repolarization of the cardiac action potential, and if it is defective, the associated condition is called long-QT syndrome, a condition that may lead to sudden cardiac death. While the gating mechanism of domain swap K plus channels has been extensively characterized, the gating mechanism of non-domain swap channels like ERG is still unknown. Since the clarification of this issue could provide a basis for a rational drug design against the long-QT syndrome, we addressed the problem with an integrated simulation network theoretical approach. A homology model was built using the EAG1 closed channel, and three putative conformations of the closed state with gating charge 4, 6, and 8 were built using stir molecular dynamics. Targeted molecular dynamics was then run to drive the channel from the closed to the open state and vice versa. The analysis of the sequence of contact breakdown and formation led us to formulate two hypothetical mechanisms. The first one is the inter-subunit mechanism, where the loop S4-S5 of the N subunit pushes onto the C-linker of the N+1 subunit, causing the extension of the 6th alpha helix. The second one is the intra-subunit mechanism, where the loop S4-S5 and the 5th alpha helix directly push onto the 6th alpha helix of the same subunit. It is noteworthy that both mechanisms underlie a dynamical communication between loop S4-S5 and the 6th alpha helix. In order to identify the most likely communication pathway, we represented our protein as a graph with edges weighted by the information distance, and then we used the Dijkstra algorithm to identify the minimal path concerning the 1st alpha helix and the loop S4-S5 with the 6th alpha helix of the pore. Finally, we used the Brandes algorithm to identify residues with the highest betweenness centrality. The approach prevalently identified the intra-subunit path, either following the route loop S4-S5, S5, S6, or the longer S4, S1, S5, S6.
Alpha-7 is a nicotinic receptor belonging to the ligand-gated ion channel family, widely expressed in human brain, with a proven role in a wide spectrum of neurological disorders, including schizophrenia, Alzheimer's disease. Therefore, alpha-7 is a key target for drug development. This poster collects the results of the structural notation of several conformations we obtained by homology modeling, plus extensive full atomistic molecular dynamic simulations. I show here typical structural descriptors whose combination describes the process from the ligand uptake release step in the ligand binding domain down to the gating of the transmembrane pore. In particular, I find very interesting two aspects of this work. First, that the process could be described by the evolution of an H-bond pattern. Indeed, I found the communication between different subunits is present in the open active state at the interface between the ligand binding and the transmembrane domain, while it is completely lost in the inactive form, so likely is required for alpha-7 to function. In other words, this provides information on key interactions that could be critical to the alpha-7 work. Second, we provide a model obtained with simulation from the spontaneous relaxation of the active form upon ligand removal, which in our opinion recapitulates the characteristics of the alpha-7 resting state, with structure and dynamic properties which are actually intermediate between the open and the closed conformation. In particular, already on the time scale of our standard sampling simulation, we can observe several debating events at hydrophobic girdle, which are quite similar to the liquid vapor transitions in hydrophobic nanopores or hydrophobic gating. In this case, they are totally pivoted by the dynamics of the residues at 6' position. So in all, these results add to our previous work on the open active and the sensitized state conformation. I think that we contribute to the structural characterization of the conformation landscape of the human alpha-7 receptor and suggest also benchmark to discriminate among conformation found both in experiments and in simulation of other channels of the ligand-gated ion channel family.
Hello everyone, I'm Johny Dimuccio, and this short video is about a geometrically induced selectivity and unidirectional electroosmotic flow in completely uncharged nanopores. Ionic selectivity in nanopores is traditionally understood to be a consequence of the presence of surface charges upon the pore wall, and it is often associated with the electroosmotic flow, the control of which is crucial in micro and nanofluidic technology. Here, we propose a different, purely geometrical mechanism to induce ionic selectivity and electroosmotic flow in completely uncharged nanopores. The working principle is quite simple. Let's consider a cylindrical nanopore drilled through a neutral insulating membrane with a coaxial cavity surrounding the central conductive channel. The system is immersed in a perfectly symmetrical electrolyte that completely wet the cavity and the nanopore. When a voltage difference is applied across the membrane, two main effects occur. Ions start to flow through the pore, while charges are accumulated at the solid-liquid interfaces. In particular, the accumulated charge depends on the voltage polarity. Indeed, when a positive voltage is applied, cations accumulate inside the cavity and anions accumulate on the pore wall, and the opposite happens for a negative voltage. The broken electron neutrality inside the nanopore will result in an imbalance between the positive and negative ionic fluxes. From molecular dynamics simulations, we find that the average accumulated charges inside the cavity and inside the nanopore are proportional to the external applied voltage, leading to a switching selectivity quadratically proportional to the external applied voltage. A major consequence of this switching selectivity is that the electroosmotic flow will be always directed from the right to the left side of the membrane. Finally, we show that this unidirectional electroosmotic flow is a parabolic function of the external applied voltage, and we derived also an analytical expression able to describe the dependence on the electroosmotic flow on the geometrical parameter in a qualitative and quantitative way.
Speech for Frontier in Ion Channels and Nanospore, FINC Science and Technology. English is not my first language and it is not my second language either. I thank the organizers for the opportunity given to me for presenting my work dealing with theoretical investigation of the molecular structure, optoelectronic, thermodynamic and nonlinear optical properties of 4,5-dibromo-2, 7-dinitrofluorescein. My poster talks about some properties of a compound also called ESNB dye, which has many applications in the fields of medicine, antimalarial drugs, estimate the proteins, phototherapy, xenobiotic, cosmetics products and food. The aims of this study are to calculate the molecular structure, electric parameters, and optoelectronic properties and to compare the performance between the RHF method and different DFT approximation, such as PBE1-PBE, MPW1-PW91, B3-PW91 and B3-LYP. A basis set is a mathematical function used to describe electron distribution. In this work, we have used CCPVDZ basis set, which is a Dunning's correlation consistent valence polarized double zeta. We have studied this compound in gas phase with the help of Gaussian 09 software for numerical simulation and GaussView 06 to design, optimize the structure and draft the input file. We have also used GaussView 06 and Microsoft Office 2013 for graphical results. In the results, we can see the optimized structure of the title compound and the table 1 content the electric parameters. Due to the large values of first molecular hyperpolarizability and dipole moment of this molecule, we think that this compound has potentials applications in the field of optoelectronic. This can be a promising material for optical applications. In all the tables of this work, the blue color means that the correlation effect of electron decrease the values while the brown color increase them, and no color means that there is no influence of correlation effect of electrons. The thermodynamic properties show that the B3LYP is the best functional to study the stability of this compound due to the small value of total energy and high value of entropy. The frontier molecular orbitals show the HOMO and LUMO orbitals on figure 2 and also the variation of the band gap energy that we can see on the figure 3. We notice the very large value of band gap energy in RHF method, which does not take into account the correlation effect of electron. The table 3 show the optoelectronic properties of the compound, which are constituted by radius r, volume v, electric field E, polarization density P, electric susceptibility chi, dielectric constant epsilon, refractive index n, and the magnitude of the displacement vector d. And at the end of the table we put band gap energy A gap. Conclusion. Briefly, we used RHF and 4DFT approximations with CCPVDZ basis set to determine the electric and optoelectronic and thermodynamic properties of ES and B dye. We note that B3LYP and MPW1PW91 methods are well defined to give the best results of our study. The correlation effect of electrons is important to determine the properties of 4,5-dibromo-2,7-dinitro-3-oxo-3-H-spiro-2-benzofuran-1,9-xanthine 3,6-dialate or ES and B dye. This compound constitute an attractive object for future studies of nonlinear optical materials, because of its high values for the electric parameters. Thanks for paying attention.
Hello, my name is Helena Kadrianka, and I want to present the work dedicated to usage of bacterial ion channels as tools for structure-function investigation. Bacterial ion channels are simplified homologs of their eukaryotic counterparts and have been widely used as a model for mammalian ion channels. However, their use is limited by homotetrameric structure, which causes a radial symmetry, while in contrast, eukaryotic ion channels assemble as pseudo-heterotetramers and display radial asymmetry. Here we report two new approaches of engineering radial asymmetry in the structure of bacterial channels that were successfully used for electrophysiological experiments. The first technique is the co-expression of different monomers of the bacterial channels, as shown here in green and blue, to induce a random assembly of heterotetramers. Assuming that the assembly of heterotetramers is formed without bias, the proportion of channel types can be determined by polynomial distribution, as you see in the table 1. This can be useful for gating studies, for example, the random assembly of wild-type notchback and its L226P mutant, seen in figure 1. This mutation causes dramatic alteration in channel gating, so homotetramers can be easily separated. The current recordings from cell contraspected with wild-type and mutant monomers exhibited unique currents at -10 mV, indicating formation of unique heterotetramers. This approach has been also used to study the role of fixed charge in the selectivity filter in sodium-calcium selectivity. As you see on figure 2, the co-expression of wild-type and Letwas mutant, that is with additional aspartate in the selectivity filter, changes calcium conductors dramatically, demonstrating that the fixed charge value of -4 is characteristic for sodium permeability, and the fixed charge value of -5 is sufficient to permit calcium permeation. However, the use of co-expression always suggests the pool of different channel types. To address this complication, we have generated concatemer, which you can see on figure 3, and which can be targeted to direct point mutations. The additional aspartate in either domain 1 or domain 2 led to induction of calcium permeability, supporting our previous observation with mixtures of monomers. The current work is an important step in overcoming limitation in usage bacterial ion channels to gain unique insight into structure-function relationship. Thank you.
Welcome everyone. My name is Bhabha Ghosh. Presently, I am a postdoctoral fellow at Indian Institute of Science Education and Research, Thiruvananthapuram, India. And currently, I am working on atomistic molecular dynamic simulation of organic molecules to understand their crystallization and aggregation behavior. So, I am also looking for my next postdoctoral opportunity. Today, I will be talking about part of my PhD work, which is on pulling a folded polymer through a nanopore. This is important in the context of polymer translocation, because polymer translocation is important for DNA, RNA and proteins to transport them from one place to the other inside living systems. This is also important in the context of gene therapy, drug delivery, DNA sequencing. So far it was believed that the movement of the polymer through these nano channels in the biological system happens in a single file way, but with the advent of modern sophisticated experimental techniques, people started to find that this happens in a folded way as well. So to understand this process from the theoretical perspective, we carried out the molecular dynamic simulations of this process by considering a wall in which a polymer is threaded with different branch lengths N1 and N2, and this middle monomer is applied with a force. The details of the simulation model is given here. In the simulation protocol, we have considered two stages, equilibration and translocation of the chain. Further, we have found out that the translocation happens in four stages of the four stages. So the first stage is the tension propagation along both the branches. Second stage is end of tension propagation in the shorter segment. Third one is the tension propagation on the second segment, while the first has already crossed the pore. And fourth is ending of the tension propagation at the longer segment. We have calculated the waiting time distribution of the monomers inside the pore. This basically tells the average time spent by the monomer inside the pore. We have calculated it for three different branch ratios: 51-51, 31-71 and 11-91. Further, we have looked into the translocation times for each of the branches and found out the new scaling relationship for the folded translocation case, and it has been found that it matches well with the MD theory and equation 14, which is there in the paper attached in this poster segment of the website. Also, we have calculated the velocity of the monomers during the translocation process, and we have found out that there is no sliding of the monomer movements. And in conclusion, we have found out the isoflux tension propagation theory for these folded polymer translocations. To further know about this, please go through my paper attached in the poster segment. Thank you very much for your attention.
Hi everyone, my name is Will Gibby and I am presenting my poster titled Physics of Selective Conduction in KCSA and Natchback, although here we'll primarily focus on KCSA. Understanding, predicting and optimizing the ionic transport properties of nanopores remains a critical challenge to both biophysics and nanotechnology. An interest is motivated by the growing importance and diversity of applications, including biosensors, water purifiers, and of course biological channels and their role in health and medicine. The aim of our work is to describe the permeation properties of such pores, and to do this we develop and derive a statistical and linear response theory. We will apply this theory directly to KCSA, and in other work we have applied it to Natchback, and we expect it to be applicable to other biological channels and artificial pores alike. Our system is a pore thermally and diffusively coupled at either entrance to bulk reservoirs. The pore itself houses an arbitrary number of binding sites, and the system an arbitrary number of species and concentrations of ions. Since occupancy of the pore can be classified into discrete states, we take the appropriate steps to derive a grand canonical ensemble describing the system and its statistical properties. Away from equilibrium, we followed the ideas of Kubo and Zwanzig to derive conductivity at each site. We find this to be proportional to the statistical fluctuations within the pore, i.e. the variance and covariance of particle numbers between ions of different species and at different sites, and by summing these site conductivities in an appropriate manner, i.e. in analogy to resistors in series, we can calculate the total current and conductivity through the pore as a function of our energy spectra and structure of the pore. For application to KCSA, we'll limit ourselves to two conducting ions, potassium and sodium. We'll take site selectivities from a reference given in the paper, and we'll compare to experimental data, again referenced in the paper, for wild-type KCSA and a mutant where the structure of the S4 site has been varied. We find the energy states to be highly quantized depending on the number, position and type of ions inside the pore, ultimately favoring potassium over sodium, as expected. Conduction occurs when there is a degeneracy between neighboring energy levels. When these are the lowest energy states of the system, and when the pore itself is conducting. Away from these conditions, we find negligible conductivity or blockade, and we find that the occupancy to be relatively fixed in integer values. Finally, as can be seen in figure 4, we see good agreement between theory and experimental data, with the mutant having a reduced current of around a 1/12 of the wild type and the occupancy of S4 being reduced. Thank you. Are there any questions?
Hi everybody, I am Alberto Bioti from Sapienza University, and my poster is about simulations of ion permeation in a hydrophobic nanopore. The system we studied via molecular dynamics simulation is composed by a solution of sodium chloride in water. Immersed in this solution is an atomistic model of hydrophobic nanopore. The hydrophobicity and the geometry of the nanopore are such that formation of a vapor bubble may occur inside it. So this pore may exist in a wet state with water inside and in a dry state with few or no water molecules inside it. By studying this system, we want to understand how different is the permeation of ions depending on the pore state. We computed the free energy of our system performing restrained molecular dynamics simulations. The free energy was computed as a function of two variables, the axial position of the ion and the number of water molecules inside the nanopore. We observed 4 minima, corresponding to the two possible states of the pore, dry and wet, and to the ion being far from the pore on the left or on the right side. From the free energy map, we can see that for this particular pore, the dry state is more energetically favorable than the wet state. What can also be seen is that the translocation of the ion through the pore is very unlikely to occur in a dry state, since there is a high energy barrier. This can be better shown comparing the free energy for the translocation of the two ions when the pore is dry and when the pore is wet. When the pore is wet, there is a barrier for the ion entering the pore, which is around 3 kBt for sodium and 5 kBt for chloride. This means that our pore, even if not charged, is selective for sodium with respect to chloride. The situation is very different when we evaluate the free energy for the ion translocation in the dry state. In this case, we have barriers around 43 kBt. This is expected because permeation in the pore, when it is dry, would mean that the ion has to lose at least part of its solvation shell, which is a very unfavorable condition. We repeated the previous computations with a voltage applied to the system. In this case, the effect of the field is such that the profiles are not symmetric anymore. Also, in the case with the voltage, we see that the barrier for sodium is lower, indicating that sodium ions will contribute more to the ionic current than chloride ions. We also evaluated the effect of the voltage on the wetting transition. We see that the effect of the voltage is to lower the energy of the wet state. This is interesting because it suggests that this kind of pores can be created by the voltage switching from a conductive state when the pore is wet to a non-conductive state when the pore is dry.
Hi, my name is Mindy Rongop. I am a first-year PhD student at Phoenix Laboratory, hosted by CNRS and Sorbonne Université in Paris. I will give you a quick overview of my work on fluctuations of ionic current in nanopores that has been done last year with my PhD supervisor, Benjamin Rothenberg. Nowadays, thanks to breakthrough in nanopore technologies, it is possible to accurately measure the ionic current through single pores. These measurements come with some noise, which can be studied through autocorrelation and spectral analysis. Because this results from microscopic effects, we believe the noise carries some precious information about the physico-chemical properties of the system. But in order to interpret it, we need an accurate model for the microscopic dynamics. We start the analytical work with the continuous mean field approach, which describes the concentration fields of each ionic species in an implicit solvent, and coupled FICN diffusion and Colombian interaction to the Nernst-Poisson equation, and add to that a white noise stochastic component that models the collisions between the ions and the solvent. From this model, we are able to get a solution in Fourier space in the linear regime, with some calculus and algebra, we now have the total charge current density in the bulk as follows. To take into account the effect of confinement in the nanopore, we approximate the boundary condition with some restriction on the wave vector of the bulk solution. These derivations come from the work of Zorkot and Gorestanian done in 2018, which we have generalized to generic ionic solutions. So far so good. We can now write the current power spectral density as follows, an integral over the wave vectors that couple the effect of geometry and the bulk solutions can now be computed numerically. Now we want to take a look at a specific case, which is a binary solution, and discuss the effect of asymmetric diffusion coefficients, something that is often neglected in the literature. We look at high and low frequency regimes and show that new behavior occurs at relatively high salinity, something that was not predicted with the symmetric case. While midfield approach are quite limited, because they do not take into account short-range interactions, we have shown that it is possible to get good insight on the electrodiffusive processes through Nernst-Poisson modeling.
Hi there, my name is Stefan and I'm postdoc with Professor Ceccarelli in Cagliari. Here I'm going to present our results on two pore channels, mainly mammalian TPC1 and human TPC2, TPC2 being very important in fighting COVID. So we are doing molecular dynamic simulations, and with MTPC1 we showed that for sodium ion passage to occur, the biggest barrier, the biggest obstacle that sodium has to overcome is hydrophobic gate, and through hydrophobic gate, in hydrophobic gate, sodium ion has to lose one water molecule in order to pass, but also hydrophobic gate has to expand, so these two meet, and as you can see here, there is a region where the expansion of the hydrophobic gate and the size of hydration shell are comparable and compatible, and then passage occurs. However, in this protein we couldn't see a passage when we apply external electric field, only when we use biased molecular dynamics. So we moved to HTPC2, which is only ligand gated, and here we observe that in cavity, which is this space between selectivity filtering and hydrophobic gate, different numbers of sodium ions can be present during the molecular dynamic simulations, and we saw that in open conformational state, the most probable state of the cavity suggests that there is one sodium ion inside. So we wanted to see what does it mean, and we ran external electric field simulations, and there again we saw that depending on whether or not we have sodium ion inside prior the passage, the different modes of passage are going to occur. So if there is a sodium ion inside, we're going to have very fast kinetics of transport with two different scenarios. So either the second ion comes while the first one in the cavity, and then the first one leaves and second takes its place in the cavity, which resembles a bit of knockoff mechanism seen in calcium channels. And also we have the situation when this ion comes inside, dwells a bit with the ion that's already there, but then leaves. And we were interested to see what does it mean. So we ran again metadynamics, fixing the number of ions inside, and we saw that the number inside that dictate the ion flux in high or low concentration. So if we have 0 ions inside, we have better fluxes. At low concentration, we have 1 ion inside, we have better fluxes and high concentrations.
Nanopore sensing is a tool toward better and smarter batteries. Batteries, as one of the most versatile energy storage technologies, plays a central role on the ongoing transition from fossil fuels to renewable energy. Lithium-ion batteries are the technology of choice, as they offer the largest energy density while their cost is continuously decreasing. Nevertheless, improvements are needed to increase their lifetime and sustainability. One issue to be addressed is the redox shuttle, the migration of soluble polysulfides between the two electrodes. Cyclodextrin trapping properties are being explored by placing them in the membranes of sulfur-based electrodes. Up to now, existing analytical techniques do not allow to detect and sequence at the single molecule level these parasitic redox species. Here, we propose a high-resolution detection of these by using a nanopore technology, in particular the resistive pulse, a protein channel, alpha-hemolysin, with a cyclodextrin adapter as a simplified model to understand the encapsulation of polysulfides by cyclodextrins and to extend smart membrane design for lithium sulfide batteries. The reversible equilibrium complexation between polysulfides and cyclodextrin was characterized by proton NMR spectroscopy. Interactions between the polysulfides and the different cyclodextrins was determined following the chemical shifts of different protons in cyclodextrins in the presence of polysulfides. Molecular docking affinity ranking over all inclusion complexes led to the same results, highlighting the stronger affinity for the beta cyclodextrin. Stoichiometry of binding was also tested for the various polysulfides, concluding that BS4 is the strongest. And this observation is consistent with a similar trend obtained by calculations. Detection of the different polysulfide species by nanopore current blockades are characteristic of the species size, sequence, and chemical modifications. We can observe a statistical decrease in the event current amplitudes correlated to a decrease of sulfur number for each complex. Calculations based on approximated quasi-one-dimensional continuum description of the nanopore electric resistance were used to calibrate the system, and they are in excellent correlation with the experimentally observed average current blocked. In summary, our findings offer molecular insights and innovative perspectives to use nanopores as electrolyte sensors for designing smarter batteries. We thank our collaborators, our software environment, hardware providers, and you for your attention. Thank you very much.
Hi, my name is Gonçalo Paulo, and I'm a first-year PhD student at Sapienza University in Rome. And this poster is about the motivation of my project, which is related to the importance of channel noise in modeling neurons. The current hallmark model for modeling the generation of action potentials in neurons, which is called the Hodgkin-Huxley model, is a completely deterministic different set of differential equations, which doesn't take into account any kind of noise in this process. When this model was first introduced, this made sense as the number of ion channels was so big and the current that went through each one of them was so small that it could be thought to neglect their effects in the generation of ion channels. Now it is known that some neurons do require noise for their normal behavior, and so studies on a single channel noise have started to be developed. Taking into account noise in the Hodgkin-Huxley model shows that increasing the internal noise of a neuron can actually increase its sensitivity, and there seems to be an interplay between the internal noise that the neuron has and the external noise from the input that the neuron receives. There's also already some models that can study the behavior of single channels and introduce Markov chains that completely mimic the stochastic nature of the channels. We are interested in the formation of bubbles inside ion channels, which seems to be a relevant process for anesthetic drugs, and we are gonna use molecular dynamics to study this spontaneous formation of bubbles in hydrophobic channels. We hope to see some results that can be applied to sensing and lab-on-chip applications.
Ion channels and membrane transport proteins selectively allow the movement of ions and water across cell membranes. In particular, the selectivity for anions still remains poorly understood. A number of crystal structures of ion channels and membrane transport proteins suggest that chloride ions may form favorable interactions with hydrophobic residues. My research explores the influence of hydrophobicity on anions at hydrophobic interfaces and in nanoconfinement. Molecular dynamic simulations have revealed that anions at water hydrophobic interfaces adsorb to this surface, as shown in Figure 2. However, this is only seen when polarizability is explicitly modeled in simulations. While explicitly including polarizability is more physically accurate, it is also more computationally expensive. Here we use a modeling method called electronic continuum correction, or ECC, that aims to include polarizability effects in a mean-field way whilst maintaining low computational cost. We use the ECC method to study anions at a water-alkane interface. This revealed chloride ions prefer to interact with the interface than the sodium ions, shown by the peak and the shift towards the interface in Figure 5a. But if we look at Figure 5c, this characteristic absorption is not seen when we do not include polarizability in our simulations. We then moved on to look at the effects of anions in hydrophobic nanoconfinement by building a model hydrophobic nanopore and still using the ECC method to include polarizability effects. Our simulations revealed the same effect as seen with water-alkane interface, where the chloride ion absorbs to the hydrophobic interface, and even when we change the salt concentration and the size of the nanopore. The area under each graph represents the number of ions in a region of the pore. So if we compare the areas under the chloride graphs with the corresponding sodium graphs, there appears to be more chloride ions present in the region, therefore suggesting that there is some chloride selectivity happening. Overall, our simulations have revealed that chloride ions preferably interact with hydrophobic interfaces, both at water-alkane interfaces and in a model nanopore. These effects are only seen when we consider electronic polarization, and we should really bear this in mind when studying biological ion channels.
Hi everybody, I'm Dalia Sun, a medical biotechnology student. I'm working under the supervision of Professor Giacomello and Dr. Carlo Guardiani. In my poster, I'm going to talk about our MD simulation study of quark channel. It's a calcium channel activated by calcium, and it's intimately related with our health and several immunodeficiency diseases. Why is this channel so interesting? The point is that it has a very controversial gating mechanism. Besides an electrostatic gating, it's thought to have also a hydrophobic gating with the formation of a bubble which is going to block the channel. So our purpose is to study the conformational change that underlies this particular type of gating. In the literature, one of hypotheses suggests a gating mechanism based on the rotation of pyrolysis, and this rotation is supposed to displace some hydrophobic residues away from the pore and let water and ion pass through the channel. At first, we analyzed the closed structure of quark channel, and surprisingly we find out that the channel radius is always large enough to let water molecules pass through, although the channel is closed. But then, by analyzing the mean force potential of water, we find that a large central area corresponding to the hydrophobic region, it has a very high free energy which will not allow the water molecules to pass through. This outcome is compatible with the hydrophobic gating. Therefore, during our trajectory, we observe the formation of a stable vacuum bubble at this region. And then, by analyzing our simulations, we actually find that the rotation of pyrolysis, particularly it occurs as the twisting with different rotation angle at different level of the pore, and moreover, when the rotation angle is maximum, we find that there is a sudden increase of the solvent accessible surface area of the pore, particularly at the hydrophobic region. So in conclusion, we speculate that the pyrolysis rotation is going to create a thermodynamically favorable environment for the bubble formation, which is typical of the hydrophobic gating. Please enjoy my poster.
Hi, my name is Emanuele Delari and I am a PhD student in Theoretical and Applied Mechanics at Sapienza University of Rome, under the supervision of Professor Alberto Giacomello, and I'm studying the slip at liquid-liquid interface using molecular dynamics. The slips are liquid-infused surfaces that achieves excellent slip and drag reduction properties by substituting the solid-liquid interface with a liquid-liquid one. The reasons of this high degree of slip is not well understood yet, also due to the fact that the phenomenon of slip at liquid-liquid interfaces still has not been deeply studied in the literature. One of the hypotheses of this behavior is the presence of a high degree of slip at the lubricant working fluid interface due to the presence of a third phase such as contaminants or gaseous phases. In my study, I address this topic and I look towards to characterize the slip behavior of the liquid-liquid interface in presence of a third phase. In my simulations, I model a true immiscible Lennard-Jones liquid system undergoing a shear flow, where third phase atoms are inserted at different concentrations and with different interaction with the liquid phases. The shear flow is simulated through the implementation of the slot scheme and the deformation of the simulation box. My results show that the third phase inserted influences the slip behavior of the system and the interface properties. In particular, it has different effects for different gas-liquid interactions, causing an increase of the slip length when they are weaker of the liquid-liquid ones, and vice versa. Although the results show that a gaseous phase can increase the degree of slip, the changes measured cannot explain the slip length observed in the liquid-infused surfaces, suggesting that this effect alone cannot justify the slip behavior of the slips. Thank you for your attention.